Chapter 12 Respiration in Plants. All of us breathe to live, but why is breathing so essential to life? What happens when we breathe? Also, do all living organisms, including plants and microbes, breathe? If so, how? All living organisms need energy for carrying out daily life activities, be it absorption, transport, movement, reproduction, or even breathing. Where does all this energy come from? We know we eat food for energy, but how is this energy taken from food? How is this energy utilized? Do all foods give the same amount of energy? Do plants eat? Where do plants get their energy from? And microorganisms, for their energy requirements, do they eat food? You may wonder at several questions raised above. They may seem to be very disconnected, but in reality the process of breathing is very much connected to the process of release of energy from food. Let us try and understand how this happens. All the energy required for life processes is obtained by oxidation of some macromolecules that we call food. Only green plants and cyanobacteria can prepare their own food. By the process of photosynthesis, they trap light energy and convert it into chemical energy that is stored in the bonds of carbohydrates like glucose, sucrose, and starch. We must remember that in green plants too, not all cells, tissues, and organs photosynthesize. Only cells containing chloroplasts that are most often located in the superficial layers carry out photosynthesis. Hence, even in green plants, all other organs, tissues, and cells that are non-green need food for oxidation. Hence, food has to be translocated to all non-green parts. Animals are heterotrophic, that is, they obtain food from plants directly, that is herbivorous, or indirectly, that is carnivores. Saprophytes like fungi are dependent on dead and decaying matter. What is important to recognize is that ultimately all the food that is required for life processes comes from photosynthesis. This chapter deals with cellular respiration, or the mechanism of breakdown of food material within the cell to release energy, and the trapping of this energy for synthesis of ATP. Photosynthesis of course takes place within the chloroplast, that is in the eukaryotes, whereas the breakdown of complex molecule to yield energy takes place in cytoplasm and in mitochondria, also only in eukaryotes. The breaking of C-C bonds of complex compounds through oxidation within the cells, leading to release of considerable amount of energy, is called respiration. The compounds that are oxidized during this process are known as respiratory substrates. Usually carbohydrates are oxidized to release energy, but proteins, fats, and even organic acids can be used as respiratory substances in some plants under certain conditions. During oxidation within a cell, all the energy contained in respiratory substrates is not released free into the cell or in a single step. It is released in a series of slow stepwise reactions controlled by enzymes and is trapped as chemical energy in the form of ATP. Hence, it is important to understand that the energy released by oxidation in respiration is not, or rather cannot be, used directly, but it is used to synthesize ATP, which is broken down whenever and wherever energy needs to be utilized. Hence, ATP acts as the energy currency of the cell. This energy trapped in ATP is utilized in various energy requiring processes of the organisms, and the carbon skeleton produced during respiration is used as precursor for biosynthesis of other molecules in the cell. 12.1 Do plants breathe? Well, the answer to this question is not quite so direct. Yes, plants require oxygen for respiration to occur, and they also give out CO2. Hence, plants have systems in places that ensure the availability of oxygen. Plants, unlike animals, have no specialized organs for gaseous exchange, but they have stomata and lenticles for this purpose. There are several reasons why plants can get along without respiratory organs. First, each plant part take care of its own gas exchange needs. There is very little transport of gases from one plant part to another. Second, plants do not present great demand for gaseous exchange. Roots, stem and leaves respire at rates far lower than the animals do. Only during photosynthesis are large volume of gases exchanged, and each leaf is well adapted to take care of its own needs during these periods. When cells photosynthesize, availability of oxygen is not a problem in these cells since oxygen is released within the cells. Third, the distance that gases must diffuse even in large, bulky plants is not great. Each living cell in a plant is located quite close to surface of the plant. This is true for leaves. You may ask, but what about thick woody stems and roots? In stems, the living cells are organized in thin layers inside and beneath the bark. They also have opening called lenticles. The cells in the interior are dead and provide only mechanical support. Thus, most cells of a plant have at least a part of their surface in contact with air. This is also facilitated by loose packing of parenchyma cells in leaves, stem and roots, which provide an interconnected network of air spaces. The complete combustion of glucose, which produces CO2 and H2O as end products, yields energy, most of which is given out as heat. Reaction C6H12O6 plus 6O2 gives out 6CO2 plus 6H2O plus energy. If this energy is to be useful to the cell, it should be able to utilize it to synthesize other molecules that the cell requires. The strategy that the plant cell uses is to catabolize the glucose molecule in such a way that not all the liberated energy goes out as heat. The key is to oxidize glucose not in one step but in several small steps, enabling some step to be just large enough such that energy released can be coupled to ATP synthesis. How this is done is essentially the story of respiration. During the process of respiration, oxygen is utilized and carbon dioxide, water and energy are released as products. The combustion reaction requires oxygen, but some cells live where oxygen may or may not be available. Can you think of such situations and organisms where oxygen is not available? There are sufficient reasons to believe that the first cells on this plant lived in an atmosphere that lacked oxygen. Even among present living organisms, we know of several that are adapted to anaerobic conditions. Some of these organisms are facultative anaerobes, while in others the requirements for anaerobic condition is obligate. In any case, all living organisms retain the enzymatic machinery to partially oxidize glucose without the help of oxygen. This breakdown of glucose to pyruvic acid is called glycolysis. Glycolysis. The term glycolysis has originated from Greek word glucose for sugar and lysis for splitting. The scheme of glycolysis was given by Gustav Embden, Otto Meyerhof and J. Parnas and is often referred to as EMP pathway. In anaerobic organisms, it is only process in respiration. Glycolysis occurs in the cytoplasm of the cell and is present in all living organisms. In this process, glucose undergo partial oxidation to form two molecules of pyruvic acid. In plants, this glucose is derived from sucrose, which is the end product of photosynthesis, or from storage carbohydrates. Sucrose is converted into glucose and fructose by the enzyme invertase, and these two monosaccharides readily enter the glycolytic pathway. Glucose and fructose are phosphorylated to give rise to glucose 6-phosphate by the activity of enzyme hexokinase. This phosphorylated form of glucose then isomerized to produce fructose 6-phosphate. Subsequent steps of metabolism of glucose and fructose are same. The various steps of glycolysis are depicted in figure 12.1. In glycolysis, a chain of 10 reactions under the control of different enzymes takes place to produce pyruvate from glucose. While studying the steps of glycolysis, please note the steps at which utilization or synthesis of ATP or NADH plus H plus takes place. ATP is utilized at two steps. First, in the conversion of glucose into glucose 6 phosphate, and second, in the conversion of fructose 6 phosphate to fructose 1,6 bisphosphate. The fructose 1,6 bisphosphate is split into dihydroxyacetone phosphate and 3-phosphoglyceraldehyde PGAL. We find that there is one step where NADH plus H plus is formed from NAD plus. This is when 3-phosphoglyceraldehyde is converted to 1,3 biphosphogyceraldehyde glycerate. Two redox equivalents are removed from PGAL and transferred to molecule of NAD+. PGAL is oxidized and with inorganic phosphate to get converted into BPGA. The conversion of BPGA to 3-phosphoglyceric acid, that is PGA, is also an energy yielding process. This energy is trapped by the formation of ATP. Another ATP is synthesized during the conversion of PEP to pyruvic acid. Can you then calculate how many ATP molecules are directly synthesized in this pathway from one glucose molecule? Pyruvic acid is then key product of glycolysis. What is the metabolic fate of pyruvate? This depends on the cellular need. There are three major ways in which different cells handle pyruvic acid produced by glycolysis. These are lactic acid fermentation, alcoholic fermentation, and aerobic respiration. Fermentation takes place under anaerobic conditions in many prokaryotes and unicellular eukaryotes. For the complete oxidation of glucose to CO2 and H2O, however, organisms adopt Krebs cycle, which is also called as aerobic respiration. This requires oxygen supply. Fermentation. In fermentation, say by yeast, the incomplete oxidation of glucose is achieved under anaerobic conditions by set of reactions where pyruvic acid is converted to CO2 and ethanol. The enzymes pyruvic acid decarboxylase and alcohol dehydrogenase catalyze these reactions. Other organisms like some bacteria produce lactic acid from pyruvic acid. The steps involved are shown in figure 12.2. In animal cells also, like muscles during exercise when oxygen is inadequate for cellular respiration, pyruvic acid is reduced to lactic acid by lactate dehydrogenase. The reducing agent is NADH plus H plus, which is reoxidized to NAD plus in both the processes. In both lactic acid and alcohol fermentation, not much energy is released. Less than 7% of the energy in glucose is released and not all of it is trapped as high energy bonds of ATP. Also, the processes are hazardous, either acid or alcohol is produced. What is the net ATP that is synthesized? Calculate how many ATPs are synthesized and deduct the number of ATPs utilized during glycolysis. When one molecule of glucose is fermented to alcohol or lactic acid, yeast poison themselves to death when the concentration of alcohol reaches about 13%. What then would be the maximum concentration of alcohol in beverages that are naturally fermented? How do you think alcoholic beverages of alcohol content greater than this concentration are obtained? What then is the process by which organisms can carry out complete oxidation of glucose and extract the energy stored too? Synthesize a large number of ATP molecule needed for cellular metabolism in eukaryotes. These steps takes place within mitochondria and this requires oxygen. Aerobic respiration is the process that leads to a complete oxidation of organic substances in the presence of oxygen and release CO2, water and a large amount of energy present in the substrate. This type of respiration is most common in higher organisms. We will look at these processes in next section. Next part will be uploaded soon. For any queries, please comment. Thank you.